Welcome to my lab. I'm Drew Collop. In today's lab, we're going to look at how to use a Coulter Counter. This is a machine that is used to count cells electronically. Here's the power button. Please never turn the machine off. We have a door that opens. We have a stage that we unlock by holding this down. You can push it down, it locks in place. You can bring it all the way down to the bottom or all the way up to the top. And make sure you're pushing that button to unlock the stage. We have a cup here to keep the probe wet. We then have two electric probes to send current through. One is inside the glass tube and one is outside. Please make sure you're always having the probe submerged in the Isoflow liquid. Over here is how we operate the device. Only press the start or stop button. You can read off the number of particles the machine counts here. We get two cups, one sample, and one blank. Here we have Isoflow. Notice we have it set to 10 mils. I don't want to introduce bubbles, so I'm going to put it at a 45-degree angle, like pouring a beer. Bring it all the way to the top. Slowly bring it down, making sure to not include any bubbles. We're going to do it twice, so we get 20 mils in each cup. It is important not to introduce any bubbles, as the machine does not count cells, it counts particles. If you have too many bubbles, they will go through the machine and they will be counted as a cell. So now I have two cups, each containing 20 mils of our Isoflow. I'm now going to add to my sample 200 microliters of cells using a P200 micropipetter. Here's the cells. I've subcultured them and saved the extra ones. Please note the cells will sink to the bottom, so it must be mixed thoroughly to get an accurate sample. I like to invert to mix my cells. Always invert just before sampling. Please be gentle to not introduce any bubbles. Our sample is now ready, and our blank is ready to go as well. It's time to wash the machine out. We'll take a 250 ml beaker, open the door, bring down the stage, and remove the cup that keeps the probes wet. Put it off to the side. We'll get some distilled water. We're going to use the beaker now to not make a big mess. We're going to bring it up and over, and we're going to spray down the front probe and the back probe with distilled water. Hopefully this is cleaned before you used it. The last person that used it cleaned it. However, don't always assume that. Now that the probe is clean, I'm going to run my blank. You don't really need to mix this, but I'd like you to get in the habit of mixing before you load it into the machine. We place it on the stage, bring the stage up until it stops, we close the door. Go over to the unit on the side, we press the start-stop button. This will now count the cells in half a mil of fluid. Every time we push the button, the machine draws half a mil up. You can watch the counts right here going up in real time. When the counts are done, the screen will change. This run gives us a count of 10. Please record that down in your lab notebook. You can then press start stop again immediately and it will count it again. However, if you press this button, the data is lost, so make sure you record down that value. We're going to run this 3 times, collecting 3 data points. You have a count of 9. One last time. What's happening is there's a small hole in the glass probe. As long as nothing blocks that hole, the voltage remains constant. However, if a cell passes through the hole, it blocks the flow and the fluid goes down. And there's a voltage pulse. So the machine is actually counting voltage pulses. It's not counting cells. That's why we want to have no bubbles. And we want to make sure we have individual cells and no debris inside. We've collected our 3 data points for our blank. We'll now do the same with our sample. Make sure you mix it thoroughly. If you don't, the cells will sink to the bottom and you won't get an accurate reading for the true concentration of your cells. Bring it up to the top, close the door, and again, press the start button. Every time the counter counts 1,000 cells, it will make a little beeping sound. (5,090 cells). Record that value down and press the button again. Once again, we're doing 3 runs. Why are we doing 3 of each? We're going to average them in case there's a discrepancy between one sample and another. You should get approximately the same value for your 3 blanks and the same thing for your 3 samples. If they're wildly different, you may want to reload the sample and count it again. 5,258. One last run. $5,369. So we have our 3 samples and we have our 3 blanks. We've now finished counting the cells. However, we do need to clean the instrument for the next person. We'll clean the outside of the probe the same way we started, by washing the front and the back probe with distilled water, getting off any cells that might be stuck to the outside of these probes. We'll then clean the inside by running our blank one time. The blank should contain no cells at all, and is a great way to make sure we clear out any cells inside the probe. I would like this value to be below 50. In the past, students have not sufficiently cleaned out the probe. This can lead to a complete blockage of the probe itself, and the machine will no longer be operational. Again, I would like this value to be as low as possible, below 50 for sure. Looks like the probe is a bit dirty right now, 48. It's below the 50, but what I'd like you to do if you get a value that's too high is rinse the probe again, and if it's still not dropping down, create a new blank and run that. We'll see what happens here. We had 48. Let's rinse the outside of the probe again. And then we'll run our blank again. $38, well below the 50. I'm happy with that. Please note there's a log on top of the machine. I'd like you to write down your name, the date, the time, and your end counts. This will ensure that you have sufficiently cleaned the device. Since implementing this log system, we have not had a single blockage in all the time we've been using it. If you write above 50 as your end count, you and I will have a conversation. We're now done. Take the blank out. Remember, the probe must remain wet, so we'll place that cup back on the stage and close the door. Please leave the machine on. We'll take our 2 cups. Please note these are reusable. Put your name on them. Write blank on one set, sample on the other. Going to rinse it out with some water down the sink. Going to leave them upside down on the paper towel to dry. Again, do not throw these away. These are reusable. You'll use them for the entire semester. Once again, please leave the machine on. Let's now take the raw data from the Coulter counter and complete the calculation to determine the concentration of the cells. We ran 3 blanks, we ran 3 samples. We obtained 10, 9, and 5 as our 3 values for our blank. For our sample, we obtain 5,090, 5,258, and 5,369. We then take the average of each of those. So we add them up and divide by 3. The average blank is 8. The average sample we have is 5,239. Now, if this was a decimal value, please don't round it off. Remember, we do not round off until the final answer. So you would keep all the digits and continue doing the calculations until the very end. So we have the background and we have how many particles are there. Remember, the Coulter counter does not count cells, it counts particles. So as long as we had limited bubbles and we broke up the clumps of cells, we can assume that each of these particles represents one cell. For the calculation, we need several other parameters. We did a dilution. We took 0.2 mils of cells, and we added that to 20 mils of my Isoflow. Please note, when we're doing a dilution, it is the part over the whole. So we took 0.2 mils of cells and added it to 20 mils of Isoflow. As a result, I have to include that volume of cells. We do the math now. 0.2 milliliters over 20.2 milliliters. We simplify that down and we cancel out the units. We get 1 over 101. So our dilution in the Coulter counter cup was 1 over 101. So to work backwards and find out what my concentration was initially in my test tube, I have to multiply by my dilution factor. Dilution factor is the reciprocal of your dilution. Therefore, my dilution factor is 101 over 1. So please note, if you modify the volume of cells loaded, or if you modify the volume of Isoflow in the cup, this will change your dilution factor. If you're counting cells and you believe the concentration to be very low, you would probably want to add a higher volume of cells. I don't see a reason to change the 20 mils of Isoflow. But please note that this value of 101 over 1 can change depending on how you set up your dilution. The other parameter we have to take into account is the volume. We want a concentration, how many cells per mil. This parameter has to do with when we press the button on the Coulter counter, the machine draws up a certain amount of fluid every time. How much? 0.5 milliliters. This does not change. I suppose you could go into the machine and change it, but we always have it at half a milliliter. As a result, we have a calculation here. We start with our average sample, we take away our average blank. This is what's known as normalizing the data. I'm taking my average sample, I'm taking away my blank. The blank is the background noise that's there. So this could be adding to the value of the sample. So we want to make sure we normalize the data. 0.5 mils, that has to do with the volume right here. And our dilution factor, in this case here, we're using 0.2 milliliters of cells in 20 mils of Isoflow. As a result, this is going to be 101 over 1. Let's now complete the calculation for this particular data set, and we'll determine what the concentration actually was. My average sample, 5,239. My average blank was 8. Divide by my volume. Here, it counts particles, but we will assume they're cells, so this will give me cells per milliliter. And if I just do this part of the calculation, that tells me the concentration of cells in my Coulter counter cup. But I don't care about that. What I care about is how many cells were in my test tube. As a result, I have to multiply by my dilution factor. 101 over 1. I do the math now, and I get 1,056,662 cells per mil. Let's round that off to 3 significant digits. 1.06 times 10 to the 6 cells per mil. For this particular sample that I use the Coulter counter to count, I obtained a concentration of 1.06 times 10 to the 6 cells per mil. Hopefully now you have a solid understanding of how to operate the Coulter counter and do the calculations. We'll see you in the lab.